Hi, I'm Peter Tonner, and today I'll be talking about our new interpretable machine learning modeling approach for genotype-phenotype landscapes that achieves state-of-the-art predictive accuracy. As many of you know, and as we'll see throughout this conference, machine learning has revolutionized the way in which we predict the effect of genetic mutation on downstream function. Shown here is a far from exhaustive collection of recently developed approaches, all of which rely on deep neural networks for the underlying model. In general, there has been a decrease in experimental costs, which has led to increased dataset size and coordinate model complexity, and ultimately predictive accuracy. But deep neural networks are not without their limitations. In particular, they operate as black boxes because it is usually not straightforward to understand why the model makes a certain prediction. This is in comparison to relatively interpretable approaches that trade off predictive accuracy for insight into how the model works. In general, most people assume that this trade-off between model interpretability and predictive accuracy is unavoidable. To address the black box nature of neural networks, an entirely new research field has been developed to explain these models. But by necessity, these explanations are only an approximation of the underlying model, and it's generally unclear when these explanations will be accurate. For example, feature importance scores, like the one shown here for different image classification labels, can overlap dramatically in terms of features relevant for making the correct classification to labels that are completely nonsensical. Additionally, these explanations can be quite fragile, and adversarial perturbations to the data that are imperceptible to humans can drastically change the feature importance scores. As an alternative to black box models that require post hoc explanations, we developed a fully interpretable model that is built on two key ideas. The first idea is that the representation learned for the effect of mutations should be intuitive. This is reflected in one of the more common explainability techniques used for black box models, where the features learned by the model are projected to some low-dimensional space by an explainability technique. But again, this is only an approximation to the underlying model, and it's unclear when this approximation will be accurate. In our case, we explicitly represent the effect of mutations as vectors in a latent space that will be learned by the model. We then assume that the effect of multiple mutations combine additively, as shown here. There is therefore no ambiguity around how our model represents the effects of mutations and requires no additional explanation. There is also theoretical work suggesting that biophysical constraints often lead to a relatively small number of dimensions along which function can be modulated. To represent this assumption, we place a hierarchical prior on the variance of each dimension of our model's latent space. This prior ensures that the model learns the dimensionality of the system directly from the data, and only as many dimensions as necessary are included. In practice, we find that datasets with thousands of mutations and up to hundreds of thousands of measured variants require only 3 to 5 dimensions in the model. Our second key idea is that while individual mutations may combine additively in the latent space, these representations will map to observed phenotypes in a nonlinear way. A classic example of this is when individual mutations have approximately additive effects on protein folding stability, but downstream function of that protein depends nonlinearly on that stability. So in our model, we assume that there will be a nonlinear surface that connects the learned latent representation to observed phenotypes. We place a Gaussian process prior on this surface, which is a statistical machine learning model over functions capable of learning any smooth nonlinear surface without prior assumptions on the underlying parametric family. Together, we combine these ideas into a model we call Lantern, which we consider to be fully interpretable because we can straightforwardly explain how predictions are made by the model, First, individual mutations are combined additively, given their latent representation, and then translated to observed phenotypes through the smooth nonlinear surface. And the parameters of the model have intuitive connections to biophysical processes, for example, protein folding stability. We first wanted to assess Lantern's predictive accuracy in comparison to other popular models in this space. So we developed a broad benchmark of large-scale genotype-phenotype landscapes and compared Lantern to multiple alternative approaches, including a one-dimensional monotonic spline model, as well as one of the most popular feed-forward neural network architectures for this kind of problem. In tenfold cross-validation, we found that Lantern equaled or outperformed these alternative approaches, including the black box neural networks. Lantern's interpretability means that we can also directly understand what it has learned from the data. For the allosteric protein Lacl, which modulates gene expression in response to changes of environmental ligand concentration, Lantern modeled the effect of mutations on the allosteric parameters EC50 and G infinity. On the left, we see the low-dimensional latent space learned by the model, with individual variants shown as scatter points and contours showing the posterior mean of the learned nonlinear surface. We found that the latent dimensions learned by Lantern show striking resemblance to biophysical parameters in an analytic model of allostery. Under the analytic model, changes to different biophysical parameters influence EC50 and G infinity in distinct ways, as shown by these individual curves. These patterns are recapitulated by each of the two latent dimensions learned by Lantern, shown on the right. This suggests that Lantern is learning representations that relate to the underlying biophysics of the system. We also analyzed a recently published dataset of the SARS-CoV-2 spike protein. We identified two distinct axes in the learned latent space that capture different aspects of the protein's function. First, there is the stability axis, which points in a direction of decreased structural stability, shown in green. This decreased structural stability also naturally disrupts the binding of the protein to the ACE2 receptor. A second axis, which we term the binding axis, shown in purple, also follows a trajectory with decreased binding affinity, but without any loss of structural stability. We found that residues associated with these different axes had intuitive connection to the spike protein structure. Residues more strongly associated with the stability axis are more broadly spread throughout the core of the protein. Conversely, those associated with the binding axis cluster closer to the region of ACE2 contact. So the representation learned by Lantern appear to plausibly relate to different aspects of protein structure that influence the spike protein's function. And it's important to note that Lantern had no access to structural data during training. In conclusion, Lantern demonstrates that there is no trade-off necessary between interpretability and predictive accuracy when modeling genotype-phenotype landscapes. But there's a lot more I didn't get into today, including how we make inference tractable, further insights that Lantern made into different large-scale GPLs, as well as novel metrics that Lantern provides about different landscapes. So if you'd like to learn more, feel free to stop by my poster and say hi. This is collaborative work with members of the Living Measurement Systems Foundry at NIST, and there is ample wet and dry lab work available for interested postdocs, so please reach out if you'd like to know more. Thanks.